Today is afternoon, so very good afternoon, everybody, and welcome you all to this 38th webinar of this plant breeding webinar series. These webinars are being organized by the Foundation for Advanced Training in Plant Breeding on a monthly basis. And today we have Dr. Boney Kuriakose, who will talk about metabolic engineering of plants using genome editing, CRISPR-Cas-based modification of the carotenoid biosynthesis pathway in tomato. The purpose of these webinars are to sensitize people to know and value the importance of plant breeding, and also to create an awareness on various aspects like technology integration, policy support, regulatory overview, farmers' comprehensions, and various other aspects required for inclusive development of crop improvement. Foundation for Advanced Training in Plant Breeding is a non-profit organization dedicated to enhance capacity building in agriculture, particularly in the field of plant breeding. Since its inception in 2018, ATPBR has made significant progress in advancing plant breeding education. We have organized 3 international conferences, 5 national conferences, 33 professional training sessions, and 37 webinars, reaching more than 12,000 individuals who spread not only across India, but in various other countries that include Sri Lanka, Bangladesh, Brazil, Italy, Nepal, Afghanistan, Nigeria, Kenya, Vietnam, and many other countries. ATPBR has recognized 18 breeders and scientists and 3 organizations for their extraordinary contribution to transforming farmers' income through their improved crop varieties and farmer supportive policies. We also have published 6 books and 3 books under Springer umbrellas, and we have 12A and ATG CSR certification, and we are eligible for conducting any CSR project. Now, I would like to take a privilege to introduce our moderator, Dr. K.K. Narayanan. Dr. K.K. Narayanan is trained as a plant breeder and molecular biologist. He's PhD from TNU Coimbatore and postdoc from Stanford University, USA. He's a farmer, founder, director of Metabolic Life Science Limited. Currently, he's the founder, director of Saika Seeds, director, CEO of Agri-Genome Lab, and also he's the founder, director of ATPBR. Dr. Narayanan is a member of several committees and advisory bodies, including several scientific task force on agriculture biotechnology of the DBT, Government of India, and the Vision Group on Biotechnology set up by the Karnataka government. I request Narayan sir to please take the floor.
Thank you, Aparna. I hope I am audible to everybody.
Very much.
Okay, good. Thank you for the very generous introduction. So friends, we already have close to 50 people who have logged in, and I'm assuming that some more people may join as we go along. A very warm welcome to our monthly webinar series to all those who have joined. Thank you very much. And I must also take this opportunity to thank Dr. Boney Kuriakose, who very graciously consented to give this talk today. So Boney, I have known him for a long time now, from the time he was doing his doctoral studies in Chennai, or Madras, it used to be called at that time. From the Madras University, he has graduated with a PhD degree. His training has all through for his undergraduate course for the master's degree program has been in plant biology. And after his doctoral studies, he went for a stint postdoctoral study in South Africa. And he has turned out to be a very good plant biologist, plant scientist. And I continued interacting with him off and on, but it got closer when I was associated briefly with this company called Agri-Genome, where Boney led a team of scientists who were involved with genome sequencing, genomics, and also significantly, he built a wonderful platform for genome editing using the CRISPR-Cas9 system, and validated that platform by editing the genes in 2 species, rice and tomato. I think what he's going to talk to us today is the work in tomato. Elaborate that in detail. So without much further ado, I hand over the virtual space to Boney Kuriakose. But before that, before he takes over, a couple of requests. One is that while the speaker is at his job, please keep your microphones in the mute mode. That is one. And number two, we will try to have a brief interactive session towards the end. So just to make it orderly, please post your questions in the chat box if you have some pressing questions, and I will try to moderate the discussion. I can pick up selective questions from the chat box and let Boney address these questions towards the end. So we can have about 10 minutes of Q&A at the end. And I guess, Boney, your presentation can go to about 40-45 minutes. Is that fine?
Yeah, that's fine.
Okay, now over to you, Boney. It's all yours.
Able to see the presentation?
Yeah, it's coming.
In fact, my special welcome before that to people from West Africa, Ibadan in Nigeria, the colleagues from NIHOT. Welcome, all of you.
Yeah, welcome to them.
Thank you.
Thank you for having us.
Thank you, Manila. We can see you fine.
Yeah.
Go ahead, Boney.
Okay. So very good morning to all of you from this side in Europe, from Paris. So thank you to the Foundation of Advanced Training in Plant Breeding for giving me the opportunity to talk about the work that I did actually in India with AgriGenome. The title is going to be about metabolic engineering of plants using genome editing, and I'm going to talk about the work that we did on tomato carotenoid biosynthesis pathway. But I will also give you an overview of CRISPR-Cas9. So it's one of the different genome editing techniques which was invented and the one which is most commonly used now. So it is derived from the bacterial immune system. Basically, it's a system by which the bacteria protects itself from the viruses which infect them, the bacteriophages. So when a bacteriophage infects when a bacteriophage infects the bacteria, they inject their DNA into the bacterial genome. It gets integrated, and within the bacterial genome, it kind of leaves small traces of these of the bacteriophage gene in this area, which is called as the clustered regularly interspaced short palindromic repeats, or which are called as CRISPR. And they get transcribed and they get transcribed into what is known as CRISPR RNAs, and the CRISPR RNAs then associate with this protein called as Cas9. These are DNA nucleases which can cut the DNA, and based on which part of the bacteriophage genome is integrated into the bacterial genome and which is transcribed. The next time there is a reinfection by the same bacteriophage, this sequence is recognized by this CRISPR RNA, and then it helps the bacteria to fight further infection by this bacteriophage. So it's a very, very beautiful system which was invented or evolved by the bacteria to fight itself from viruses. But then the wonderful team of Jennifer Doudna, Emmanuel Charpentier, and others, when they discovered it, they also thought that it could be now engineered for using genome editing. And that is how the whole process of genome editing using CRISPR-Cas9 came into the picture. There are other genome editing methods which were previously employed using zinc fingers, talents, etc. But none of them were so sequence specific and so simple as the CRISPR-Cas9 method. And that's why the CRISPR-Cas9 became like a very, very common way of doing genome editing. So if you are able to see this picture where I've made a close-up of how the CRISPR-Cas9 complex would look like. So if there is a genome, then there's like a complementary RNA part, which is part of the guide RNA and complex, which goes and binds there. And then the whole guide RNA complex is bound by the Cas9 protein. And the Cas9 protein is the native Cas9 protein is has the ability to cut both strands of DNA. So wherever there is binding of the complementary RNA sequence, as well as on the opposite strand. So it makes cuts about 3 bases from what is called as the PAM site. The PAM site is basically for this particular Cas9 enzyme, it's a sequence of NGG. So it can be any nucleotide followed by 2 GGs. It is usually about 3 bases from the NGG, and that is where the cut is made. And when this cut is made, it basically breaks the DNA into 2. And in any organism, there is a repair mechanism of DNA repair happens using different repair mechanisms. So one of the repair mechanism when there is a double strand break is called as non-homologous enjoining. So basically, the different ligases try to come together with different polymerases and tries to repair if there's a break in the DNA. And during this break, which is highly error prone, there can be a lot of errors which happen during this because it's a double strand break and it's trying to repair itself. Sometimes there are a few bases which are deleted, sometimes a few bases are inserted, sometimes large deletions happen, and so on. So this is made use in the most common way of doing a CRISPR is when you design a guide RNA to a gene and it goes, it makes a double strand breakage, and when it tries to repair, it either introduces a nucleotide or deletes a nucleotide, and there is a mutation introduced because of that, and that mutation can result in, for example, if it is within a protein coding sequence of the gene, it can introduce a frame shift leading to a non-functional protein or a truncated protein in many cases. It has also been used for inserting DNA. For example, if you design a guide RNA which makes the double strand cut, but you can then introduce any foreign DNA with a homologous sequence to the area where you expect the breaks to happen, then this DNA which you can externally provide can get integrated. And this is one of the ways for introducing genes or making corrections in genes by assisted recombination. So these are the ways by which the CRISPR-Cas9 can be used for making the changes. It is very common now all over the world. People are using CRISPR-Cas9 for various applications from metabolic engineering of yeast to human gene therapy to plant metabolic engineering or plant disease resistance engineering, and so on. Classic examples are where there's been like a lot of publication on the bacterial blight resistance in rice, where people have worked with the genes which are responsible for knocking out the genes which are responsible for making the rice susceptible and thus making resistant rice varieties. So the different mutations which happen can be classified as has been classified as SDN 1, 2, and 3. And based on this, the whole global regulatory network in different countries have been, all the regulatory bodies have been like, in the last few years, trying to bring CRISPR into more into application mode and try to bring in the regulatory framework for approval of organisms or plants which have been engineered with CRISPR. So the most simplest one is where there is it's called as a SDN1 mutation, where it is like a double strand break. It gets repaired normally. There is a single strand insertion, single base pair insertion, or a single base pair deletion. It is very common. It happens in nature, and when it happens in nature, nobody notices it unless you go a few generations down and you have to sequence it, and then you found, okay, there is a single nucleotide polymorphism, or SNP, which has been introduced. So it's very common. So if you are able to do that using CRISPR-Cas9, then it's considered the most straightforward, simplest form of a gene knockout in many cases. And it's called as SDN1. SDN2 is when you can introduce a very, very short repair using a very, very short template which you can introduce, which is not basically, it's if there is like a small change in 1 or 2 amino acids that you want to make, you just change that region. So it's also not very complicated. If you want to introduce a whole new gene, or you want to repair like a whole large region of a gene, then it becomes SDN3, which is in many countries also considered equivalent to being a GMO or transgenic. So other than that, in many parts of the world now, if you look at this global map, you will find that in many parts of the world, if there is no transgene in the plant that you use for engineering, if there is no, there's been only SDN1 or SDN2 changes, then they are like mostly considered as non-GMO plants in most of the world, except Europe. Unfortunately, Europe is still stuck with kind of treating them as GMOs, but you can find that there has been a huge advancement in gene editing research because of this in the rest of the world. India especially has been like in the last few years made drastic changes in its regulatory network, and today India is one of the countries where there is approval, but it's based on case by case. So it's based on what traits are and what crops, etc. So coming to this presentation, my presentation is going to talk mostly about what we did in the early part of this decade, around like 2020, 2021, when gene editing in India was still very new. At AgriGenome, we were working on establishing a platform for genome editing in plants. So for that, we made some choices, and how we went about doing it is going to be in the next few slides. So the objective was to establish a genome editing platform, obviously. So we wanted to work with a very commonly known crop, and so one of the reasons why tomato was chosen. We also chose a South Indian variety, which was chosen because there was literature on its tissue culture and transformation. So it was a very good choice. And the third choice was to look at what we wanted to do with it. And so we wanted to modify the carotenoid isomerase pathway. So the carotenoid biosynthesis pathway in plants, it's a very, very long pathway. It starts with the phytoin synthase, and then the phytoin desaturase, and then comes into the different isomerases, and then before it goes into the lycopene biosynthesis and the carotene biosynthesis. So lycopene is the red pigment that you see in tomato. It is known for a very high antioxidant activity. You you find lycopene capsules in pharmacies the world over. So the gene that we chose, CRTISO, or the carotenoid isomerase, is a gene which converts tetra-cis lycopene, which is also called as prolycopene. So prolycopene is converted into all-trans lycopene, and this all-trans lycopene then goes into the keratin biosynthesis pathway. So our objective was to see if we can knock out this gene, carotenoid isomerase. The reason being that there has been earlier studies, there has been reports of mutants of this gene giving a different color in tomato fruits. So there's been different reports of different genes, different mutations in the carotenoid isomerase leading to this phenotype called as the tangerine phenotype. The tomato fruit looks almost tangerine in color, and even the flower is different. So we wanted, we thought this would be a very good candidate for the proof of concept study, because if we are able to knock out this gene, then it would stop the conversion of prolycopene, which is the tangerine color, into all-trans lycopene, which is the red pigment. So tomato PKM1 is a very, very dark red tomato phenotype. It has, it's a very popular variety grown across South India, has flat fruits, has very good storage, keeping capacity basically, so suitable for long distance transport, and also used for by farmers. So now, how do we go about engineering or making the constructs for doing the knockout? So we looked at the gene structure of the carotenoid isomerase in tomato. So it's a single copy gene in the whole tomato genome, which is good because if there are more than one copy of the gene present in a genome, then there is a chance that when you knock out one gene, the other gene might compensate, and so you will not really get a true phenotype. But because it's a single copy gene, if you knock it out, then the chances of you getting the phenotype is more or less certain. It's a gene which has, it's a big gene. It has about 13 exons and 12 introns. The total gene size is about 5 kb plus. The protein that it makes is a 616 amino acid protein. So one consideration when we make mutations using CRISPR-Cas9, or for that matter, any site-directed mutagenesis, is to knock out the initial part of the gene, so that, you know, if there is a mutation introduced in the first few bases of the gene, then it would definitely lead to frame shifts, which can give rise to a stop codon somewhere very close by, and so which would mean that you will never get like a functional protein. Whereas if you try to do it at a you know, at a distal end of the gene, too close towards the end of the coding sequence, sometimes it might still make a protein which is functional, so you will not get the true phenotype. So we designed three guide RNAs, two of them in the first exon. So this is just the amplified picture of the mRNA. This is the first exon. So we designed two guide RNAs on the first exon. And we also designed a guide RNA on the 5' UTR. So the objective was that if you design guide RNAs to a coding sequence, it is going to most probably knock out the gene. So if it knocks out the gene completely, then in many cases, you will see a phenotype. For example, carotenoid isomerase, you're going to see a phenotype of color change. But some of these knockouts can be very lethal, and you can end up in having drastic phenotypes which for research purposes might be good, but if you're looking at a product development or if you're looking at a crop improvement program, then it might not be well suitable. In which cases you could actually try to knock down. So instead of doing a complete knockout, you try to reduce the amount of gene expression and try to keep it a little lower than what is usually found, so that there is a reduced phenotype, not like a complete knockout phenotype. And for that, you can choose either the promoter region, or you can use the 5' untranslated region where you can introduce mutation. So we wanted to try that as well. So we designed one of the guide RNAs to the 5' UTR as well. And a very commonly used vector for doing transformation of guide RNAs into dicotyledonous plants is PKC401. So we cloned the guide RNA under Arabidopsis U6 promoter, and this vector also has, following the guide RNA cloning region, it has the guide RNA scaffold region, and then it also carries the Cas9 coding sequence driven by a CAMV35S promoter. So and then you can select the plant using kanamycin resistance as the selection. So this was the method which we used for making the vector. So we made the vector and then transferred them into Agrobacterium tumefaciens LBA4404. LBA4404 is one of the commonly used Agrobacterium strains for transformation of plants. And for tomato, what we did is we used 10-day-old PKM1 seedlings. So we surface sterilized the seeds, we put them for germination. The seedlings, when they're 10 days old, you cut them, cut them into smaller pieces, and then you infect them, and then you select them on kanamycin for about 2 weeks. In about 2 weeks, you will start seeing shoots emerging from the hypocotyles, and also from the different young stems as well. And in about 4 weeks, you have developed shoots, which are then moved to a proliferation media, and then in about 15 days, you start seeing rooting in tomato. So this is the transformation method which we followed. And at the end of the transformation, we were able to obtain a number of phenotypes. So the plants, because we were not very sure what the phenotype was going to be, we grew the plants to its full growth stage, flowering as well as fruit development in the greenhouse, and then we were able to see different phenotypes. So you can see, like, there are some couple of fruits which were tangerine in color, and then there was also others which didn't look very different. So then we did the sequencing of the regions where the mutations happened, and we found that most of the mutations we found were in the UTR region, except in one where we found a mutation in both the 5' UTR. So just to go back one step, for the transformation, although we designed different guide RNAs and cloned them into individual constructs, when we did the transformation, we mixed all of them together. So the idea was that you wanted to have all possible combinations of mutations happening. So we did not transform them individually for the UTR or for the gene part. So because of that, in plant ID 1 or the line 1, we found that there was mutation in the 5' UTR as well as in the exon. There was another plant number 42, which gave us like a very clean tangerine phenotype. And so we started then looking at that in more detail. So when compared to the wild type plant, you could find that the flowers were slightly smaller. They had a more paler appearance. In terms of the plant, also the top leaves of the plant were more yellowish in color. There was some effect on the chlorophyll biosynthesis as well. The fruits at different stages were smaller, and they also had a different color as they grew and as they became ripe. So we then selfed these plants in T0 because we wanted to get to a homozygous phenotype soon. So after selfing in T1 or the next generation, we were able to get fruits. Sorry. So we were able to get the same phenotype in the next generation as well. You can see that compared to the wild type, the flowers are more paler. You have a more yellowish phenotype on the top leaves as well. So we then went ahead and did sequencing to see what was the mutation. So what we found is that line 42 actually was a biallelic mutation, which had on one of the alleles, it had an insertion of an A in the sequence. So if you look closely at the sequence, it is GGT, GAT in the wild type, but in the mutant, it is GGTG, and then there's an A insertion, and then it becomes GAT. And in the other allele, there was a deletion of G, so it is GGTGGA, and instead of that, the G is deleted, and you have like GGTGA. So it's segregated as per Mendelian ratios. We were able to get some plants with an A insertion, some plants with a D deletion of the G, and we also were able to get biallelic mutation in a number of plants. We did the sequence analysis of the protein to see what was the effect of the mutation. So we actually mapped the mutations, tried to map the mutation on the coding sequence, and then did a translation to see. So if there is an A insertion, then because of that, there is a stop codon introduced at about 120 amino acids. And if there was a G deletion at about 127 amino acids, there was a stop codon introduced. So both the mutations resulted in a truncated protein, a non-functional protein, and that is why we were able to see this phenotype, which was tangerine in color. One of the important considerations when we do any genome editing is to look for off-target effects. So generally, when you design the guide RNAs, there are different tools, online tools, open source tools, which are available for common crops where the good genomes are available. So you can always screen the genome for potential off targets, and then you can also design primers to sequence those regions to see if there has been any off-target effect. For this plant, we were unable to see for the most common off-targets. There were no changes in the potential off-target regions. So we went ahead and then wanted to characterize. Okay, we saw the effect. We saw there's a change in the color. Now, does it correlate to the metabolite? So the metabolite that we are going to look here is all-trans lycopene. So all-trans lycopene results in plants. The all-trans lycopene results in fruits which are like dark red in color. So we did a HPLC analysis of the fruit pigments, and we found that if in the wild type, you can see this is the lycopene peak, which is present in all the different wild types. Whereas if you look at the mutant, in the mutant, the lycopene peak is completely absent, and there is other peaks which appear, which are the precursor isomers of all-trans lycopene, and they started accumulating more, and that is why we are seeing the more, because of the pro-lycopene mostly accumulated, you are seeing this color. So it was very clear that, okay, the conversion from or your carotenoid isomerase function has been completely affected, and there has been very less or no conversion of the pro-lycopene to all-trans lycopene. So it was observed in both the different alleles, homozygous plants of both the alleles, as well as in the heterozygous. Because the lycopene biosynthesis pathway, the carotenoid biosynthesis pathway extends itself to other pigments like xanthophylls, and also in the chlorophyll biosynthesis. We looked at the plants to see if there is there's been a difference in the content of those metabolites as well. So we did assays for total chlorophyll as well as for xanthophyll and carotene. And in the plants where the number 42, where we saw like a yellowing of the leaves, we found that the xanthophyll and carotene content was lower in the leaves, as well as the chlorophyll content was lower in the leaves. So we then did a further analysis of the carotenoid estimation in the leaf, as well as also in the fruit and the flower. And so in conclusion, for the number 42 plant, we found that, okay, it doesn't accumulate all-trans lycopene, it accumulates more of the precursor, which is the pro-lycopene, and both in fruits, leaves, as well as in all the different, although it was a biallelic mutation in both the alleles, when it's aggregated, when it became homozygous, the trait was retained. So it was very interesting for us that it happened that way. And then we also continued working with the other lines, especially line 58. Line 58, we saw that there was a large deletion, inside in the 5' UTR region, which was about 193 base pairs. So just to go back to the gene, we designed a guide RNA to the 5' UTR region. So in the 5' UTR region, we were able to get this big deletion, which was about 193 base pair. So when you do the sequencing, we are able to see that compared to the wild type, there's a large deletion of the 5' UTR region. Interestingly, these plants, they look very, very normal to the wild type in the first generation of the T0 plants. The reason was that this deletion happened only in one strand, and it was a heterozygous in one of the alleles, and so it was still heterozygous in T0. When it segregated, when it became homozygous, and when the 193 base pair deletion became homozygous in the T1 plants, we were able to see there was a reduction in the amount of pigmentation. It was not as drastic as when there was a total knockout, but there was a reduction in the total amount of all-trans lycopene. So basically, what has happened is that there is no complete knockout because the protein is still functional, but because there has been some elements in the 5' UTR which were critical for the expression of the gene for synthesis of the RNA, it has been deleted. So the transcription of this probably seems to have affected. And we also did the same analysis for the chlorophyll estimation, the carotenoid estimation, and also the HPLC for the so you can see that this is On top is, on the right side is the wild type, which has about 65.2 milligram per kilogram of all trans lycopene. In the homozygous, the other allele, which is same as the wild type, we are seeing almost the same amount, whereas in the homozygous, where you had the 193 base pair deletion, you're seeing there is a reduction. It's only about 45, and there's also like the other peak which is appearing. So how did it affect the gene expression? So this is something which we wanted to look at. So we did quantitative RT-PCR studies on the biosynthesis pathway from leaves as well as fruits. And what we found out that, okay, in number 42, there is only a single base pair change in the coding sequence. There is no change in the UTR or the promoter region. So basically that means that the transcription happens. The transcription, although the RNA might have become less stable, the transcription happens, and there is still transcription you can see for the number 42 in both leaf as well as in fruit for the carotenoid isomerase gene. Whereas in number 58, you can see that number 58.7, there is almost zero transcription happening for the carotenoid isomerase gene. So we also compared it with another gene in the pathway and saw that the other gene's expression was maintained, whereas the carotenoid isomerase in the UTR knockout was like almost zero expression happening. So it shows that there is a reduction in the expression of the gene. So in summary, what we found out was that we are able to do genome editing using CRISPR-Cas9 in tomato. So this was at that time one of the first reports from India on genome editing in plants. There was a previous report in banana, but in phytoindisaccharase, but other than that, there were no other publications at this time. So we designed and developed all the different tools which are required for doing the CRISPR-Cas9 expression mediated genome editing. We were able to make a knockout as well as a knockdown in tomato successfully using CRISPR-Cas9, which were directed towards either the coding sequence or to the UTR region. Similarly, we did some work on rice also. I will quickly run through a few of them. In rice, we chose a starch biosynthesis regulator. So we wanted to knock out a transcription factor because it affects a lot of genes downstream. So we wanted to see what would be the drastic effect of that. We did similar work. We designed guide RNAs. We made the vector. We did transformation of rice. We got different lines of rice which had the mutations. And then we are especially interested in one line where we saw that there was a single base pair A insertion, and then we grew the plants in T0, similar to what we did for tomato. We grew them to T1, and in T1 we looked at what was the effect of the rice on the protein. So we found that the mutated protein was again truncated at the whole protein was about 512 amino acids. The mutated was only 300 amino acids. The amylose content, because it was a starch regulator biosynthesis, starch biosynthesis regulator transcription factor. So we looked at the amylose content, and there was like slight changes in the amino content for the rice. Interestingly, the seeds were slightly smaller than the ones of the wild type, and the plant also was slightly smaller. So this is another thing. We couldn't continue the study for long, but this was also a part of this, how we established a platform for, built a platform for genome editing. So we did it for both tomato as well as for rice, rice being more of a food crop, and a monocot, which is also used as a model plant of study. So if you want to do knockout studies, rice is also like one of interesting crops to work with. With that, I would like to end. I would like to thank the team at AgriGenome, my erstwhile colleagues, and also late Dr. Subhash, who was part of the tomato transformation. He is the one who established, helped us establish the tissue culture and transformation of tomato. Dr. Josh Thomas, Dr. Shekhar, Sam, and also ATPBR for giving me this opportunity to present the work, Dr. K.K. Narayanan. Some of my colleagues from Genomines, where I am currently working with, has also joined. I would like to thank also the Genomines management, because they're a startup and we don't get many opportunities to talk and interact with people, public, and talk science. So this was a good break from it. Thank you very much.
Thank you.
Thank you. I think you can take off the slides from the slideshow.
Yes.
So see, the claps are coming. People are appreciating your talk. And thank you very much for a wonderful presentation. We are almost 50 minutes into the presentation, and there is time for some discussion, interaction. But before that, we have the former colleague of Boney also joining us, Dr. George Thomas. George Thomas, you are also part of this work. You want to add something to what Boney said?
Not really. I think Boney has covered everything. It was.
He has done a wonderful job.
He has done a wonderful job, and it has been a pleasure and an honor working with him. Yeah.
So let me look at the chat box. There doesn't seem to be major questions. Boney, of course, I am wondering if you have this. You may not have the slide for this, but can you?
Krishna has raised a hand. Okay.
Sorry.
Somebody has raised a hand. Two of them.
Sriram Kumar. Sriram Kumar, are you there online?
No, Trishta Padwal and Naima. Two of them I can see they have raised hands. Can you see them?
Can you unmute yourself and then come with your question?
Trishta Padwal, can you hear? You seem to have raised your hand. Naima.
Anyway, there is one question now in the chat box, Boney. I think it is, you have knocked out the lycopene content. Is it possible to knock a gene and then get high lycopene content?
Yeah,
that was going to be my question.
Okay.
So
Satish has asked this question.
Yeah, please.
I think it's a very, very, very interesting question, very relevant question as well, because I think this is what people are trying to do by doing the knock-ins. But if you probably add like another copy of the gene and expressing it would look like another transgenic to me. But yeah, I mean, you can knock in a gene, but you can not knock in a gene completely, because you need the whole expression system there. You have to have a promoter, a terminator, and so on. So yeah, you can do it, but at the end of the day, I think in many places it would still be considered a transgene introduced.
Let me add to that, Boney. I think this is about another nutraceutical molecule, GABA, gamma aminobutyric acid.
Yeah.
The first commercial transgenic or gene-edited product is a tomato line called Sicilian Rouge, which was introduced by a company called Sana Tech. The name of the tomato is Sicilian Rouge. It was introduced in Japan, which is otherwise so much against gene modification, but they have permissions for gene-edited lines. There, they have knocked out a gene and increased the accumulation of GABA. The GABA accumulation is controlled by certain negative regulators in the genome. So they knocked out the function of the feedback inhibition so that GABA continues to accumulate, and those tomatoes are providing more health benefits. So that's just for the information of people. Yeah, go ahead. Now there is Hassan Kolkas who has asked a question. Thank you, Boney. Did you plan reintroducing a functional modified version of the same gene to check if the gene by itself is responsible for the resulting phenotype? Now, how are you sure that your knockout is responsible for? So I think that's the import of the question.
Yeah, I think if you go back to what I told at the beginning, there was only a single copy of the gene, and the genome of tomato is one of the best completed genomes. So you know that there are no other copies to compensate for it. So if you knock out, and you know that the carotenoid biosynthesis pathway again is like one of the best studied pathways in plants in general. So yeah, I mean, ideally, you can do that by like overexpressing another copy of this gene and then complementing it and showing it. But that was not part of our study. Our study was mostly to show that we can knock out gene and bring in a phenotype.
I see somebody has raised a hand, but I'm not able to see them.
It is Naima Sosarai.
I think Pamela has raised a hand.
Pamela and Naima.
Hello. Can you hear me?
Yeah.
Yes.
Yeah,
Hi,
thank you very much.
Go ahead.
Yeah. Thank you, Bonnie, for the clear and very nice explanation. Sorry, can you hear me?
Yes.
I think,
Yeah.
yeah,
Please go
okay.
ahead.
So thank you, Bonnie. Everything was super clear and very interesting for me. I just have a question, like out of the curiosity, that after the you made the tomato lines, the knockout lines, if any other project is made to make the, like breeding projects to continue with the lines that you made to make a like homozygous line out of the breeding, or if yes, like how many generation you did the breeding, and how was the project?
So right now, okay, so go back to the historical part of it. So we were working continuously. We were at, I think, the T2 generation, but at that point of time, so I have moved out from there. So I'm not sure about the progress of it, but I think yeah, it is. We were able to show that it has been like inherited until the second generation.
Oh, okay, perfect. Thank you.
Thank you.
Thank you.
Pamela had a question. No?
Okay, yes, I have a question. Yes.
Yeah, go ahead, Pamela.
Okay, thank you very much for that very clear and beautiful presentation. I just wanted to ask, can you share some of the bioinformatic tools that you used for screening off-targets, potential off-targets in tomato? If you can just share some of your bioinformatic tools that you use for that.
Okay.
Then the second question again is, when you did those large deletions of about 193 base pairs forming heterozygous system, I was just wondering, can those large deletions be classified as SDN3, or is there like a limit to how many deletions you can do to be termed as large deletions for the 193 base pair at the promoter region?
Okay. So to the first part of the question, I think we have many tools, web-based tools, which are available, especially if you're working with crops where good genomes are available. You can use tools like CRISPR plant There is one called CRISPR-P, which is from a Chinese lab. There's also CRISPR, CRISPR. So these are CRISPR is something which we are using currently in our in the work that I'm doing in my current job. But for this particular one, we were using CRISPR-P for the design. For the second part, about the 193 base pair deletion. So I want to say that when you do CRISPR, when you're doing targeted mutagenesis, which result in homolog, what you call as non-homologous end joining repair, there are chances that you can have very drastic deletions sometimes, and this was one of an example. We did not intend to make the 193 base pair deletion. It happened as part of the knockout process.
Let me
So
interrupt here. I think her question was also, how big, what is the limit of the deletion to classify it as SDN3?
yeah, unfortunately, I don't know the answer. I don't know. Maybe you know.
I guess a few base pair will still be SDN2, but anything more than that, is SDN3. It may be a little bit of a subjective assessment by the regulators.
Okay.
Pramila, I guess you are satisfied
Yes.
with the answer.
Yes, I am. Thank you very much.
I must also add, Boney, that Pamela and her team are trying to establish a gene editing system in the NIHOT campus. They have a wonderful facility there in Ibadan. And I think, Pamela and your group, if you have any more questions, you can send it email to us and we can pass it on to Boney. And I'm taking the liberty of saying that Boney will be happy to respond. Am I correct,
Yeah.
Boney?
Sure, yeah, no problem.
Yes, and
Yes,
we'll also
Suren,
be
I
glad
think
if you can
can get
share,
on.
Can I
if you
ask
can
one
share this
question?
publication as well. We'll be grateful to have this publication shared
Yeah,
with us.
I can share it with Dr. Aparna.
Thank you very much.
Go
Okay,
ahead, Suren.
Yeah, I had a question. Thank you very much, first of all, Boney, for that very, very interesting presentation. I'm a tomato guy for all my life. So you talked about, some genes for color, I have worked on some of them. Like OGC is called Old Gold Crimson. It's a single recessive gene. So I have always been thinking that if gene editing can insert also is coming under the SDN 1 or 2, right? So
if it's not
OGC
more
is
than
a single recessive gene. It's a single recessive gene. I forget the chromosome number, probably 6 or I don't remember the chromosome number now. But similarly, there is high pigment gene, HP. They're all recessive genes, which could help in converting, say, a tomato line which doesn't have great color. But if we do this addition, so would that be considered GM or would that be gene edited?
So I think if the base pair changes are not more than a few, it is still considered as SDN 1 or 2. It will not be considered as DNT, but 3. But if you are like introducing a sequence which is larger, then it would be considered as SDN3, and then it would go under the GMO approval system.
Okay, thanks.
Let me add to that bit, Suren. Some of the natural mutants have been analyzed by sequencing, and there have been examples of using gene editing to create the similar mutants. For example, the ALS gene, which confers resistance to sulfonylureas in rice, for example, it's a natural mutant, but some companies have also done gene editing to create that something that is similar to natural mutant. How will the regulatory agencies consider that? Will it be normal breeding or something is yet to be seen, I guess. But if it is 1 or 2, I would think that if I were the regulator, then I would pass it off as SDN1 or SDN2, not more than that.
Yeah, because a single recessive gene, very easily transferable, but it will take at least a few years before it is transferred.
Okay, I think we have time for 1 or 2 more questions. I'll take one, which is by Dr. Nagarani Maharajan. She has written, are there any long-term stability concerns for the gene edits in successive generations of tomato plants?
Generally speaking, it is like any other mutation. So once you go into further generations, it gets stabilized and it should be there. You are also, if you are using a transgenic system, you generally want to segregate out the vector inserted there, so that you have now the mutations in your lines and they are stabilized. So that way it would be considered like any other SNP in any other plant. It just goes into the further generations.
Okay, so another question, I think you can respond to it with reference to your experience in tomato and rice. What is the success rate of knockout using the CRISPR-Cas system? This question is by Joseph Musaija. I guess he is from Ibadan or Nigeria.
So with respect to success rate, it is always dependent on how.
What is the percentage of wildlife to mutants you get? I think that probably gives a.
Okay, so I think it depends on how efficient your transformation system is established in the particular variety that you're working with. In some varieties, you get better transformation. So the better the transformation, the higher the chances of knockouts happening. So in tomato, rice, in varieties where transformation systems has been very well established, you will get very high, like 10% or above knockouts. Sometimes you will get about 15-20% transformation rate, and then you get 10% knockout rate. So that is the minimum that you can expect in. But as you go into crops which are less studied, where transformation systems are still being established, it's not as efficient, then you might not get that good efficiency.
Good. Okay, we are almost 1 hour and 5 minutes into the presentation. Unless there are any pressing questions, I would like to close this meeting. I will just 1 minute for anybody with any pressing question.
Just one more, not a question, but like a request. We had discussed earlier about how both institutions can collaborate, and in terms of capacity building, is there any way that this opportunity can be opened to us? Because we need to build more capacities within our system in order to establish a genome editing pipeline within NIHOT in Nigeria.
Sure, Pamela, I think it is possible, but let's take the discussion offline. Is that okay with you?
Yes, that's fine. Yeah, that's fine. Thank you.
So thank you so much, Boney. At short notice, you agreed to give this webinar. It was wonderful, and it was so crisp and logically taken through. So wonderful. I'm sure all the listeners would have benefited from this. On behalf of all the participants in this, let me put together my hands to give you a big hand. You can see my hand here.
Thank you. Thank you.
Thank you.
Thank you. Thank you very much. And thank you. Back to you, Aparna. I think you can give us closing comments and close the meeting.
Yeah, thank you, Dr. Boney, for giving a wonderful presentation. And all the applause are coming in an emoji form as well. You can see that participants really like the presentation. And if any question come across later, I hope you don't mind to respond it if I send you via email. So is it okay with you if I send you an email with any questions come across?
Yeah,
okay.
No problem.
Thank you very much for all your time. And thank you all the participants for joining us for this wonderful lecture. And we will see you again next month with another exciting lecture. Thank you, everybody. Have a very nice evening and nice weekend to all. Goodbye to all.
Thank you.
Thank you very much. Bye-bye.
Bye.
Bye. Thank you.